Hi, I'm John Watt, and welcome to Ever Wondered, the show that takes you behind the scenes to look at what's been achieved at the cutting edge of New Zealand's scientific research. This week, we dig deep into archaeology and paleontology to discover how New Zealand evolved and where it might be heading in the future.
I'm James Crampton. I'm a paleontologist at GNS Science. Paleontology is the study of fossils. Any dead animal or plant that was alive millions of years ago. Paleontology often gets confused with archaeology. Archaeology is concerned with the human remains, and paleontology is anything that's sort of pre-human, so older than a few 100,000 years or a few million years. So there's different type of paleontologists. There's macropaleontologists, I'm one of those, and we study large fossils, so typically things like fossil shellfish, but also, if you're lucky, fossil dinosaurs and plesiosaurs and all those things. Then there's micropaleontologists, which they study fossils that are microscopic. And in fact, for every fossil shell or fossil bone we might find, there's tens of gazillions of microscopic fossils. Limestone and chalk are rocks made completely from the skeletons of single-celled microscopic fossils. Typically, we tend to work with microscopic fossils now, simply because you can find millions of them in a small piece of rock.
Microfossils are important, as certain types can tell us about the history of sea temperature and the change in ocean currents, helping us to design climate models for the future.
New Zealand actually is a great place to do paleontology for a number of reasons. Firstly, New Zealand sits in this sort of glorious isolation. The shellfish that live around New Zealand, about 90% of them, 90% of the species, are only found in New Zealand. So people think the sea's all connected and these things would just get around, but in fact they don't. So the fossil history can tell us all about how these things have evolved in isolation over millions and millions of years. Between 23 million years ago, New Zealand was almost entirely submerged. This sort of fossils are found right up into the middle of the South Island, right up close to the Southern Alps. And so what it tells us is that these date from that time when New Zealand was almost entirely underwater.
All this rich information is stored in a unique database named FRED.
FRED is the Fossil Record Electronic Database. New Zealand's the only place on the planet that has a database of almost every place where fossils have been found in the country. And having this information on a computer allows us to answer questions in a way that no other country really can.
Now all these fossils have to come from somewhere. Let's meet a scientist who's getting his hands dirty on dry land to discover what was in our ancient oceans.
My name's Ewan Fordyce and I'm Associate Professor in the Department of Geology at University of Otago. I'm a paleontologist, so I study fossils, and for my serious research it's mostly fossil vertebrates, backbone animals, particularly whales and dolphins and penguins. I've been really interested to look at the ancient baleen whales, essentially the ancestors of living whales like minke whales and blue whales. And I'm interested in how big they were, how they lived, did they feed on small plankton just like the modern species did, and so on. So we know that the world can change in a physical sense, and I'm really interested in knowing how the physical changes in the world might have triggered the evolution of whales and dolphins.
Ewan has located a limestone quarry in South Canterbury, abundant with marine fossils more than 21 million years old. Hard to believe that all this was once under 100 metres of seawater.
If we look around here, we might find other vertebrate bones, maybe bits of penguin, shark's teeth, odd whale bones and so on. It's a last frontier, just out there waiting for us to find. So we've got a scatter of bones here. We're not quite sure what's out on this end, not quite sure at that end, but we can say this is about our layer. So I'm going to get the chainsaw and just cut down to that layer and we'll lever out the blocks. Yeah, you've got to remember, PhD in paleontology, you've got to be able to use a pick and a shovel.
This site is still an active limestone quarry, so the race is on to get as many bones out as possible before they are crushed by the machines to sell as fertiliser on farms. I want to be a paleontologist. Look at all the cool stuff you get to use. Ewan uses heavy-duty equipment to cut the surface rock down to the fossil layer. Once there, small precision tools are used to prevent damaging the delicate bones. The entire block is then wrapped in a plaster cast in order to transport the treasure safely back to the lab.
Mostly what you're getting rid of there is tiny, tiny fossils. I could see them when I was looking through the scope. Little microfossils, less than 1 millimetre in diameter.
They just look like grains of sand.
The ancient whales seem a lot more primitive in terms of their structure, but what intrigues me is there was actually a lot more diversity in terms of major lineages than there seems to be today. So modern day whales and dolphins seem to have a lot of species in just a few lineages, whereas if we go back into the geologic past, it seemed there were probably more different lineages, perhaps each with its own cluster of little species.
Studying our distant past allows us to better understand the forces still at work on the Earth today and help us to predict the changes that are still to come.
If we want to understand climate change and what it might do to the world, one way is to use these hugely complex computer programs called climate models. The other way you can try and understand that is look back through time to find times in the past that might, in one way or another, be similar to what we think might happen in the future. And you can see how climate behaved in the past. And so using these two methods together, we can try and understand climate change.
I think working on these fossil vertebrates is all part of human endeavour to understand the past, where the ancestors of modern animals came from and when they arose, and perhaps to give us a better understanding of how they and us fit into global systems.
Let's go meet an archaeologist who is looking at the relics of early human settlement to help us better understand our interaction with the environment.
My name is Dr. Marian Turner. I'm a lecturer in archaeology at the Department of Anthropology at the University of Auckland. My field of interest is New Zealand and Māori archaeology, and I have a number of research projects, but one of them is currently the excavation and investigation at Tapu Tiki Tiki Pa in Waiuku. We don't often get to excavate pa because they are very significant sites from both a heritage perspective, from iwi perspective, and in this case, the pa is getting damaged by the trees.
This big scoop here has been caused by those trees falling down there, and so you can see they are taking out huge chunks of this pa.
So with permission from local iwi, Ngāti Te Ata, the archaeologists set to work.
We get involved in people's rubbish, what people have left behind. It can tell us a lot about their day-to-day patterns.
So this is what us archaeologists look for, where there are breaks and exposures in a surface. And here we've got basically shellfish. We've got cockles, and they're usually in quite a dark soil because of the cooking process, the charcoal used to burn the wood. Sometimes the oils make it quite greasy.
We sample pretty much everything. Like we sample soils, we sample the fills, the materials that are in the various features, any food remains, the charcoal. All these different elements and materials will provide a variety of information or specific types of information.
After meticulous gathering of all the materials, it's back to the lab at Auckland University for the long process of sorting and analysis.
More samples for you?
The XRF machine uses X-rays to look at the geochemical makeup of an object or material like stone. From this, they can match up the geochemical signature, which will then tell them where the materials came from.
From that sort of information, we can work out the interactions between people at that time, who the people at the pa may have had contact with on a regular basis.
So what's this artefact here? It looks quite tiny and fine.
It's the smallest adze in the world. It's Nelson Marlborough argillite. It's from South Island, Nelson Marlborough, D'Urville Island, possibly.
So what does it mean that you found this all the way up here in Auckland when it originates from Nelson?
Because it's high value, it was distributed all around the country. The more valuable the material, the further it went. But this is probably made from a flake off a larger adze. There are a number of reasons why the pa is of value to us. There is the research aspect, of course, there is the heritage education aspect of it, and there's the preservation aspect of it. In order to understand our present and where we're going in the future, it's nice to know where we've come from and what the story is in the past. I mean, that's our general mission statement.
Marianne's research offers us great insight into the past lifestyles of human habitation in New Zealand. Now let's meet a scientist whose research looks at the human impact on the marine environment.
I'm Ian Smith, an Associate Professor in Archaeology at the University of Otago. Much of my research is focused on finding out what animals people exploited in the past, what that can tell us about what people were doing, but also what that can tell us about those animals in their environments and the way in which they've changed over time. One of the things that archaeology does tell us is that Maori did have an impact on the environment around them. Most of their food, in terms of animal food, was coming from large animals, particularly seals. In some parts of the country, moas were very important. The moas became extinct during the time that Maori were here, and it's almost certainly the case that the major cause of that is hunting by people. Fur seals were breeding right up to the north of the North Island, and after about 200 or 300 years of Maori exploitation, there were no fur seals left in the North Island. And over time, fish become increasingly important, and shellfish as well become a really major source of food. When Europeans arrived, almost all of the fur seals that were left in New Zealand were wiped out by commercial hunters hunting them for their pelts. By the late 1870s, the first legislation to protect fur seals was brought in, and since that time the population has gradually recovered. Particularly over the last 20 years, they've been recolonising areas. They're now turning up in places where the last time they were seen was 300 or 400 years before Europeans arrived. Using the archaeological record, we can predict where fur seals are going to return to. So one day, we'll find fur seals are breeding all around the coast of New Zealand.
So what does Ian look for to help him uncover the story of New Zealand's ecological past?
A midden is a rubbish heap, and generally it is the waste from the foods that people have prepared. That nitty-gritty stuff is actually really important in terms of understanding how people lived and how they impacted on the world around them. People typically harvest the biggest things first.
Right.
Where you've got shellfish being harvested at the same place continuously over a long period of time. So you've got a number of different layers in a site. What you typically find is that the bottom layers, they're bigger, and over time they get smaller.
Smaller and smaller.
Crayfish are a really interesting example. There's really only one bit of a crayfish that survives, and that's the little beaks or mandibles around their mouths. The size of those mandibles is directly related to the size of the crayfish. In the early Maori sites, the crayfish are about 3 times the size that you ever see today. So they used to be huge.
So we know in the past that human exploitation has impacted New Zealand's marine environment. So how does that compare to the impact we create today?
Snapper were really important for Maori right throughout the prehistoric period, and they were really important for early Europeans when they arrived. About 50% of all of the snapper that have ever been harvested have been taken out of the sea in the last 60 years. The scale of it has changed out of this world in the recent past with what you might call industrial scale harvesting technology. I think archaeology is really important in terms of contributing to how we understand the world we live in today.
We're off now to meet some scientists who are using cutting-edge technology to delve deep into the secrets of our past, using a fossil's own DNA.
Current research has been on the moa and the kiwi. Birds on isolated islands in particular lose the ability to fly. They've no longer got predators chasing them, and if they forage along the ground looking for food on the ground, there's no big advantage in flying. They're not going to another island, they're staying on their island, and they often get bigger, and if they get bigger they lose the ability to fly anyway because they are too heavy, the wings are not strong enough. The moas and the kiwi and the emu and the ostrich and the rhea, we call them ratites. Even more interesting, they're related to the flying birds, the tinamou. These South American birds are within that group as well. They have not lost the ability to fly, they still live out in the open and they can escape. They still have predators around. The relationship between the moas and the kiwis and the ratites generally with the tinamou is done from the DNA sequences. So that meant that flying was part of the normal ability of their common ancestor. And we're getting that with 2 sets of data. We get it from what we call the mitochondrial DNA that comes just from the mother's line, and we get it from the general DNA that comes from both mother and father. So both types of data are giving the same answer, and we always like it when we get the same answer from two independent data sets.
But what happens to flightless birds when new predators are introduced to the island? Lara Shepherd is looking at the impact made on kiwis since humans first arrived in New Zealand.
The birds I'm concentrating on at the moment are kiwi, and I'm also doing a little bit of work on weka as well. So we're using DNA sequencing from ancient bones, so from about 3,000 years old to a lot more recently, to look at sort of the past levels of diversity and distribution of diversity in kiwi. It's quite difficult to get DNA out of ancient bones. It really depends on how they've been preserved. So cold, dry environments are best for DNA preservation, so like a nice dry cave. But even then we only get maybe 30 to 40% of the samples to work. So here we have a map of New Zealand showing where kiwi occur at present and where we sampled kiwi bones from areas where they're now extinct. So you can see that there's large areas of New Zealand where kiwi don't occur anymore. Great spotted kiwi and North Island brown kiwi have fairly similar distributions in terms of the range, whereas roe and tokueka, which are both South Island species, they've been greatly reduced.
So through farming and things like this?
Yeah, forest clearance, fire, predation by introduced animals, that kind of thing. Yeah. Probably the most significant finding was roe. So this is a species that currently there's only about 200 or 300 individuals and one population on the west coast, this little orange dot here. They're one of the most endangered kiwi species. And you can see this actually occurred right up the west coast of the South Island and into the North Island, which we weren't expecting at all. The really interesting thing to do now would be to extend this to other New Zealand species, which are greatly reduced in distribution or even which are extinct, so we can really understand what New Zealand's diversity was like in the past.
But what happens when you look so far back in time there are no fossil records to be found? Let's meet a scientist who is looking for the origins of life using our very own DNA.
I'm Leslie Collins and I work here at Massey University, and I am a bioinformatics research consultant and a research science in evolutionary genomics. I am looking at our very, very distant past. So I'm looking at not just the ancestors of, for example, all animals. I go back even further. I go back further than the ancestor of animals and fungus. So this is between the good old mushrooms that we eat and us who eat them. We have a common ancestor. How do we look at the past when we have no fossils? We go to molecular fossils. So we go to the DNA and the RNA that are in the organisms that live today. They carry with them relics of their past. All life hangs on to the successful genes in its DNA. So the concept is, if it ain't broke, don't fix it. So what we look for are the genes that are common, that are still around between all life, and we essentially mark those as our molecular fossils. So I'm looking at Giardia lamnia, which is a little organism a little bit like this. This is the only Giardia I ever want to be holding because it causes very, very nasty intestinal disease. But from an evolutionary point of view, it is a very small genome. It has thrown out genes it doesn't need to use. So where we find genes that are common between Giardia and, for example, between humans, we're looking at very, very ancient past.
So how does Leslie go about sorting through that huge amount of data?
So this is where bioinformatics comes in. We then have to take that jigsaw and get some order into it and find the information that's locked in it. There is quite a long process involved in getting the sample to a very high quality stage and then running it through a very expensive machine and getting an awful lot of data. Next generation sequencing, instead of just sequencing one gene at a time, you're sequencing all the genes all at the same time. That data starts off from the machine as being images. Now these images you could compare to being a starry night. Those images are then taken through a computer program and converted into the DNA that we're looking at, and then we end up with our million-piece jigsaw puzzle to put together. Essentially, this is our archaeological dig.
Okay.
We are looking for our fossils in this dig. So all you can see is here a lot of letters and numbers.
Looks like gibberish.
It
to
looks
be
like
honest.
gibberish, and in fact, these are our jigsaw pieces we have to put together.
Okay.
So what we do now is we take the information we already know about, and we map that information to the information we already know about.
Okay.
And what we're looking for is differences. So we take the cellular structure of humans, of plants, of giardia, and all the other organisms with a nucleus. We've found that the cellular structure is not all that different between them. And so we can assume then that the cellular structure of the ancestor hasn't changed a lot with a lot of organisms that we see today. The sort of sequencing information like I'm using has been used in medical research, in agricultural research, horticultural research and environmental sampling. New Zealand has really taken the next generation sequencing on board in a big way. So we are sequencing kiwi, we are sequencing all sorts of different bacteria that are never found anywhere else. We're sequencing in such a way that places overseas go, ah, you're trying to do that? So we really push the boundaries in New Zealand as to what we get out of this technology.
Due to both our isolation and the brilliant work being done here by New Zealand scientists, we have a unique place in the fields of archaeology and paleontology. That's all we've got for this week. If you like the science and you want to find out more, visit the Ever Wondered website. See you again next time.